ages, and that the speed with which this organ ages actually is dependent or can be determined by probably several molecular mechanisms. And I'm really convinced that the endocannabinoid signaling system is one that is an important contributor to brain aging. And you mentioned this, it's quite possible actually that the low level exposure to THC is beneficial in old individuals. It's definitely not beneficial in young individuals, because everything we test actually seems to impair cognitive function and produce adverse effects, but in old individuals it could be positive. And I think actually the mouse is a pretty good model organism to study this, because doing these animals honestly with old mice is still a major problem. It's very expensive, takes a long time, but it's comparison to many other species, I think, very well suited. So just want to thank my collaborators. Most of this work was actually done by Andreas Zimmer and Viola and Sophie, together with Anastasia and Kerstin. All the expression profiling studies were done with Jochen Schulze and his co-workers, Astrid Rahun, and we collaborated with our colleagues from the Hebrew University on some of the chip analysis. And I just want to show you a picture of our lab, what we do when we're not working. Thank you for your attention.
Just to say that they are very impressive, excellent talk and very impressive effect of the low dose of THC. But I have a very naive question. What happened with the tolerance for THC? With the chronic treatment, because this is already well reported.
Yeah, we have actually looked at tolerance development. I probably have the data somewhere in my computer, but not in this presentation. It's interesting. The development of tolerance is much quicker or more pronounced in young individuals than old individuals. So old individuals, they do produce tolerance against THC, but not nearly as fast as young animals. So if we treat young animals for a few days actually with relatively low dose of THC, so 10 milligrams already show a good effect, we see much less in old individuals. Yeah.
What do you think at what stage of age should the THC treatment in humans?
That's a good question. Asking whether I should start smoking. Honestly, I don't think we can. I don't think it's possible to correlate the animal experiments with with human experiments. What we should do, THC is available as an oral mucosal spray. It's used for the treatment of spasticity effects of multiple sclerosis. Also in Canada, it's used as an analgesic drug. So we could use it actually to perform clinical studies, but I would first start using it with really older individuals, like people who are over 80, who have maybe some form of mild dementia. I think that would be the best score. And then you have to see actually, you really have to try this out. Yeah.
Yeah, because if I understood well, in your experiments you implant you start the osmotic pump after the birth.
Yeah.
So it's quite because at first, during the first day, the mouse is totally dependent on the mother. So maybe for a translational point of view, it's quite difficult to say, okay, this period is just related to when a human can start to smoke, to use the drugs?
You see, early in the early developmental time points, actually, we can correlate the mouse postnatal development quite well, actually, with the human development. So, as far as brain maturation is concerned, we know when cortical pruning is happening in humans. We also know when this is happening in mice. We know when they become sexually mature. All these things we know, and we can correlate. However, as the animals grow older, now the question is, a 12-month-old mouse, is it like a 50-year-old human, or is it like an 80-year-old human? There it becomes difficult, I think.
Have you tried to enhance the endocannabinoid tone, like, for instance, inhibiting the degradation of 2-HG or anandamide, and to see if you could mimic the effects of the low dose of THC?
These are exactly experiments that we're doing right now. So we're using FAR inhibitors in order to try to produce the same effects. Absolutely. These are ongoing experiments. So what I showed you is not published. This is really relatively new data.
Because is there any decrease in the endocannabinoid tone
Yeah,
in
there is.
12 of
Yeah, there is a decrease in endocannabinoid tone.
in both?
In both. So treating the animals with an inhibitor of the metabolizing enzymes for the endocannabinoids is a very reasonable strategy, actually. Maybe even better than giving THC, because it would enhance the endocannabinoid tone. Yeah.
Yes, so for the chronic 3 milligrams per kilogram infusion, either during or after administration, did you look at CB1, whether or not it was desensitized or downregulated?
No,
by
we
chance?
didn't. We wouldn't expect this. The dose is so low, actually, that we wouldn't really expect to see downregulation with such a low dose. But we haven't looked at it so far.
Can you look to the effects of cannabidiol? You know that now cannabidiol for clinical trial with the PhD has very, very promising results.
Very promising,
so
yeah.
Probably for this target could be interested cannabidiol or just the combination like Satiles, cannabidiol plus plantainide.
We actually got now from GW 3 different preparations. One high THC, one even cannabidiol THC, and one low THC high cannabidiol. And we're doing the experiments now with these 3 treatments. Yeah, would be very interesting. Yeah, the striking effect in epilepsy. Actually, interesting. But we don't really know the molecular mechanisms of the cannabidiol.
About this color intake. Have you seen a weight gain in those stage in treated young and old mice?
In old mice, we have checked this. No, THC did not in old mice produce a significant effect in weight gain. In young mice, the increase in weight, I don't know if the difference was significant or not. I have to look it up.
Is it possible though that the changes you see in improvement cognition and presumably improvement in neuronal connectivity is due just to better metabolism, brain metabolism, rather than very refined signals?
Well, how do you make the control experiment for that? We would have to find a way actually to improve brain metabolism if this is what THC is doing in some other way, to see whether this has an effect or not.
Should like block as
but that would probably have many other effects. This is
I don't understand why do you think that brain metabolism will improve? Because you look at THC, you administer THC is going to produce insulin. Well, you enhance the activity of the endocannabinoid system. You produce insulin resistance. You increase glucose levels, you increase fatty acid levels, but just because you are decreasing the capability of the body to consume glucose and fat. In contrast, if you give rimonabant, was on the market for treating obesity, you decrease glucose level, you decrease fatty acid level, you decrease cholesterol, okay? Because you are improving the consumption of this nutrient, improving the consumption because you increase insulin sensitivity. So if you extrapolate, well, this you should expect is that if you enhance cannabinoid tone, you are going to impair the capability of the brain to consume glucose. Just looking the metabolism data. You increase glucose levels, okay, but you decrease the capability of the brain to consume glucose. So I would not, in principle, I would not expect an improvement of brain metabolism, just the opposite, if any effect.
yeah.
You said that you found some differences in the transcriptome new opioid receptor. Do you think that this effect also that you saw that might be mediated by GABA is also something to do with the differences in the new opioid?
We're actually doing these experiments now with GABA knockout mice. And there is some evidence actually that it's indeed receptors on CB1 receptors on GABA neurons that are important. We see reduced effect at least of THC in GABA specific CB1 knockout mice. But we haven't completed the experiments with the glutamate specific mice yet.
Yeah, so for the early adolescent treatment with THC, have you looked at any experiments looking at opiate reward or alcohol reward by chance?
No. We haven't actually. We have not done these experiments, but this would be interesting. Actually, we would believe actually that in adult animals it affects opiate reward.
Regarding the increased risk to develop schizophrenia after THC exposure, have you found some molecular correlates or something to develop in rats or to treat these?
No, it's not. I think it's too early actually to think about drug development. This is also not the aim. So the aim would be actually to educate young kids not to smoke dope, because it increases their risk to later on get psychiatric diseases. Our aim is actually to understand how CB1 signaling contributes to postnatal brain development. This is what we want to do. And this is clinically relevant, we hope, because it's a problem, but it's more an academic problem that interests me. So I really want to know what is happening actually, which neurons really respond to changing levels of endocannabinoid signaling during this period. And it's also interesting for me to know actually what is endocannabinoid signaling doing. So we know it's very high in this critical developmental period, and I want to understand actually how this system is actually used to drive the maturation of the brain. And the long-term consequences are just an indicator for me that really something is happening. Yeah.
So do you have any insight as to what you think is happening in the glutamatergic cells? What the function of CB1 is in those neurons?
Oh, I mean, behaviorally it's clear. There's many effects. But in terms of aging, this is very puzzling. It's the opposite of what I have expected. So I stopped speculating. I don't know. I really thought it's the glutamatergic neurons that are important for this. I thought that maybe glutamate toxicity or something is important. I don't know.
Do you think that could be a relation with the CB1 receptor expressed in GFAP, right, in astrocytes? That maybe they have something to do also with aging?
Certainly. I mean, the modulation of glial cell activity could certainly contribute. We know that the GABA specific knockout, they don't recapitulate all the aspects of the complete knockout. So CB1 doesn't seem to be important for the aging and glutamatergic cells, and we don't know which other neuronal cell populations are important. Astrocytes is very good candidate, actually.
But you said that there is an increase in neuronal loss in GABA CB1 knockout mice, right?
Yeah.
Not in but have you checked the cell type?
Which neurons
if you
are
have
lost?
a preferential loss of glutamatergic cells or mainly by neuron?
No, we haven't done it actually. This is something that we can do. It's just an enormous amount of work because the stereological analysis by itself is very difficult after nuance staining, and then you have to do it for certain cell populations. So far, we simply haven't done it.
Because if you could stain with that 65 or gum kinase, then it might tell you more cues about the effect of CB1.
Yeah, no, we have to do it. It's, I need more people. But you're right, it's doable, it's a straightforward experiment that gives us some important answers. But trust me, it's an enormous amount of work. Many sections, many images to analyze. Yeah.
I have a more broader question. So it might be not good to start smoking while you're old. If it increases your sensitivity or vulnerability to addiction in general, do you plan to study that? Because you see effects in all animals on addiction vulnerability.
See, honestly, I think addiction in old individuals, and I now think about people who maybe older than 70 years or so, it's not really a problem. It's my feeling. When you reach a certain age, other problems become more pressing than the risk of cannabis addiction. In young kids, of course, I mean, this is a critical problem. And one thing that we have not addressed today is whether or not THC is a gateway drug. I believe it is, because we know it changes how we respond to other drugs of abuse. There's a long-lasting effect. So I would strongly advise actually teenagers not to smoke dope.
But that will be in both youngs and in all decades.
So in old individuals could also be a gateway drug. As I said, if you are 70, if you suffer from mild cognitive impairment, you have other problems.
With regard to old people, I think that there are two drugs, alcohol and nicotine, that can represent a problem. Because imagine well, yes, the situation of alcohol, people, I would say that it's probably the only drug where there are a certain number of individuals that start with DSM-5 criteria of dependence at late ages. It could be probably the only one, alcohol, no? But also nicotine, remember, people that start smoking, whatever, 20, 30, 40, and that then start with the epoch, with the pulmonary problem, 70, 75, and still smokes. So I think that with these two, of course, cocaine, heroin,
not a problem for people because most of the consumers die before or they have before. But alcohol and nicotine still think that even in old people can represent a major problem.
Yeah. So I don't think old people, do they start smoking when they're old?
No.
But they could increase actually alcohol consumption and nicotine consumption. Absolutely.
We know that in the Parkinson's disease is the imbalance between.
Could you speak louder?
Yes. We know that in the Parkinson's disease, the balance between the glutaminergic system and the cholinergic system will be disturbed. Could you see any symptoms of the Parkinson in this case?
No. No, we have looked for movements and also exploratory activity. No, we don't. Interestingly not. Yeah.
So do you think the ability of the cannabinoid system to modulate dopamine signaling, does it change during the lifespan? Is that?
Does it change you mean the activity of the dopaminergic system? Does it change during the lifespan?
So I think that's a possibility, but also the ability of the cannabinoid system to
modulate it.
Does that also change?
Yeah, that could be. I mean, that could account for the effect that we see stronger effects of CB1 antagonism in young animals than in old animals. Absolutely. So we cannot attribute this at the moment actually to the modulation of the dopaminergic system. Because as far as I know, nobody has performed microdialysis comparison between young and old mice. But that could be absolutely a mechanism. So we would expect actually to see decreased induce of dopamine release in old individuals compared to young individuals. Why don't you do it?
Yeah, for sure.
Okay.
So we will start now the student session. So Excel in this session. So I insist that please